The purpose of sharing these comments is to talk about ways to accelerate cancer research and to provide a context into the complexity of the challenge. If you saw the Earth from a vast distance, you'd know something about it. You would know that it rotates around the sun, that it was about 93 million miles from that sun, that it completes a rotation in about 365 days, and you could make some prediction about what elements would likely be found on that planet. If you got a lot closer, you'd discover that the Earth has an atmosphere, and if you knew that, you could surmise that the balance of oxygen and the density of the atmosphere could support organic chemistry and complex molecules. But if you really wanted to know what's going on the Earth, you'd also want to look at the signaling system. And this is an image of the, in Ukraine, the cell phones in a public gathering. And switching to human biology, humans have 23 pairs of chromosomes, as we know, 46 in total. And each chromosome is actually a long thread of DNA. As you can see in the slide, as you unravel one of those chromosomes, you could look all the way down to the base pairs. And along that long thread are segments of the DNA, which we call genes, of course. And one of the topics we want to talk about is the topic of epigenetics. And what happens is that toxins, chemicals, diet, drugs, and other factors can alter gene expression without changing the genes. And when this happens, we call it epigenetics, and this explains why identical twins often don't get the same chronic diseases or the same cancers. Epigenetic factors cause chemical reactions that interfere with gene expression without changing the sequence of the gene, so it's not a mutation. And this is important in the context of cancer because obviously people sort of think that if we could just characterize cancer genes, then we'd know how to solve the problem of cancer. It's just not that simple. So what's going on in the rest of the cell? There's a cellular signaling network that's in the rest of the cell, and the genes and the epigenetic factors essentially provides an instruction set, and the signaling system is kind of the enlivenment of all that, which makes the cell actually do the work of the cell. So what does that cellular signaling system look like? Well, we've tried to provide a bit of an image to show in this way the genes and sort of this wraparound idea of epigenetic influences, and then in the context of the cell, this vastly complex array of signaling networks does the work of the cell with trillions of molecules interacting in billions of reactions across thousands of chemical pathways with hundreds of overlapping control systems and feedback loops. And we'll talk more about this as we go along. But the point is that there's complexity at the gene level, there's complexity at the epigenetic level, and there's massive complexity at the cellular signaling level. So I was trying to make the analogy here that when you could know something about the Earth from afar, and that begins to tell the story, but it's not the whole story. And as you get closer and you see the atmosphere and know about the atmosphere, there's some analogy to that being like knowing about epigenetic factors, but there's still more. And then if you really want to know what's happening on the planet, you've got to look at the signaling system, and that's true for the cell as well. So for complex cancers, if you knew all about the genes, you'd know how to treat cancer, right? Well, not really, because epigenetic changes can alter gene expression without causing mutation. Okay, so if you knew about the epigenetic changes, would you then be able to know how to treat cancer and effectively manage cancer? Well, the answer is unfortunately no, because the reaction of the cellular signaling system to genetic, epigenetic, and local microenvironmental factors can independently drive mechanisms such as metastasis or resistance to cancer treatments. So the following slides are an attempt to explain why we see such complexity in the cellular signaling system and suggest new approaches to the treatment of complex cancers. So cellular signaling, it's more complicated than we think. Now to start off this part of the discussion, what we're going to talk about is models of cell signaling systems, and these are fairly simple models that have come about over the last 40 years, let's say. An enzyme drives a chemical reaction, sort of in this picture. The enzyme would be represented by the green arrow, number 1, and that changes protein A to protein B, and then another enzyme might drive a a second green arrow to transform protein B into protein C. And that's kind of like how a metabolic pathway might work. And then what we know is that as there's, in these kinds of an example or models, as more C is produced by the system, protein C could block the initial enzyme and shut down the process. And this is a classic feedback loop. Cellular signaling models today are based on the idea that each reaction or box, as in the picture, follows its own sets of rules, and the overall system is kind of the sum of the parts. But that's not really probably how the system works. And what we want to talk about is, this is a picture or an image of more advanced representation of interactions and interplay of a more complex cellular signaling system, which is more complex and it exhibits different kinds of behavior than what the classic model would predict. So using overly simplistic cellular signaling models to address questions in cancer research may be similar to applying Newtonian physics to questions in astrophysics, where at the end of the day you just don't get the right answers. And so what we want to do is make a couple of examples, show a couple of examples to explain why an aggregation of apparently simple cellular signaling components creates functionally emergent behavior in the cellular systems. And again, this whole discussion is intended to come back to the context of complexity and accelerating cancer research effectiveness. So in this slide, we just want to first note that very simple systems can often exhibit emergent behavior. And so we're going to walk through a very simple model, and the idea is that you could have a system which simply has two rules. And again, this is just a conceptualization to show how complicated something behaviorally can become with very, very simple rules, which will be an analogy to a simple pathway in a cellular signaling system. So here are the two rules. Stay close to your two nearest neighbors and stay away from all others. And again, what we're going to look at is a modeling software tool that can throw 30 random points onto a computer screen as a blob of points and instruct each point to follow those two rules over and over again about 15 times a second and see what happens. So here we go. I'm going to go to the next slide here and I'll click into it to make it move, but what we'll see is 30 points that come up at random, and then each of those little blobs each of those little points is following two rules. Stay close to your two closest neighbors and stay away from all others. And the exercise is to sort of see what that produces, and the idea is that through these very, very simple rules, systems can organize themselves or essentially create its own emergent behavior. And that's obviously the game here, and what you're going to see if it kept going was a circle. It'll complete in a second here. So what we see in this kind of a modulation or this kind of a modeling system is that you can, with very simple rules that have nothing in those rules said anything about forming a circle, but that's what happens. That's an example, very simple example of emergence. So in cellular signaling systems with thousands of reactions that simultaneously share dozens of components with overlapping control systems and feedback loops, it's not surprising that unexpected behaviors are going to be emerging. And so that's the kind of the first point in these cellular signaling systems. We have these models which are, again, based on early and simple concepts of protein A goes to B to C, and there's a feedback loop, but there's all kinds of emergence that's really going on underneath that. And this may, in part, explain the unpredictable resistance that's seen in response to targeted therapy to cancer as cancer treatments are given and rolled out. Another aspect of cellular chemistry that may drive complexity in cellular signaling is explained by fast-slow reactions that share common components. And the background there comes from Alan Turing. Alan Turing was famous for cryptology and decoding enigma, computer science, artificial intelligence, and mathematical biology in the 1940s. And in his work in mathematical biology, he predicted the existence of oscillating chemical reactions in 1948. About 10 years later, the first oscillating reactions were actually demonstrated in the lab. So what does this all mean? In classic thermodynamics, if you have a hot water source plus a cold water source, you put them together, you get warm water. That's sort of, again, hot plus cold equals warm is not a big surprise. Conceptually, if adding hot water and cold water resulted in an oscillating chemical reaction, you can imagine the result might be a glass of water that alternates between hot and cold instead of simply being warm, as you can see in the kind of the diagram there over time. Thankfully, the combination of hot and cold water does indeed result in warm water. But Turing's work in mathematical biology did predict that there'd be these oscillating chemical reactions. And so the following slides demonstrate an oscillating chemical reaction where the parallel fast and slow chemical parts of the reaction that share common molecules and constituents with other parts of the pathways drive these a surprising sort of non-traditional thermodynamic model. So here we are in this Briggs-Rauscher iodine oscillator reaction, which combines three different solutions. The chemistry is not important, but the three solutions are combined, and a bunch of different kinds of reactions are going on inside there. And when those chemicals are mixed, then the reactions take place. And what you see here in this little demonstration is an oscillating chemical reaction that goes back and forth as the different elements of the solution move back and forth in these fast-slow reactions. So the point here is not that a cell has this specific kind of reaction or anything like that, but that this is a model of how things in cells might well actually truly work. And the presumption that they, again, follow some basic, more simple model of A going to B going to C, like we saw in that initial primitive model, probably just isn't how the real world works. And I'll note that the spinning thing you saw at the bottom of the solution there was a magnet that's used to assure that the mixture is well mixed. So the conclusion to this part of our presentation is that it's likely that emergence and large oscillations in chemical reactions across time and reaction scale characterize the ecosystem of cellular signaling, and that traditional models of cellular signaling are vastly oversimplified, and that new tools are needed to address the issue of complexity in cellular signaling, and we think that's probably going to end up being computational modeling. Another conclusion at this point is wanting to mention that there are other things that add to the complexity of the cellular signaling ecosystem. I'm not trying to be exhaustive here. Certainly another one of the others that's out there relates to the microenvironment that's around a cancer cell or any kind of cell, and the idea is that the microenvironment, the level of oxygenation and other kinds of chemicals that are coming through a cell membrane, has significant impact on cellular signaling by upregulating or downregulating various microenvironment sensitive pathways. And oxygen and levels of oxygenation is particularly important, likely in the microenvironment interaction and cancer. In the future, we believe that more advanced study of cellular signaling will reveal that there are patterns in cancer cell reactions to targeted therapy, and that these patterns will likely suggest to us that the treatment of complex cancer will not be successful with just isolated targeted therapy. And it's probably also true that it's true that way both for isolated cancer cells and also for highly heterogeneous tumor masses. And it's also, we think, likely that complex cancers will have sequences of targeted therapies in order to drive cellular systems into reactive zones where resistance to treatment or alternative pathways cannot prevail. And we're going to look in more detail at this in these subsequent slides. So in the following slides, we're going to use the analogy of neighborhood streets to present the idea of cancer pathways, targeted therapy that blocks a part of the pathway, how resistance to treatment likely emerges, or at least can be conceptualized, and how sequenced targeted therapy may be an essential step in an effective treatment of complex cancers. And again, this is meant to be a simple analogy to help understand the concepts. So a cancer cell, in theory, is a cell that has an active abnormal pathway that results in uncontrolled cell growth or failure to have apoptosis or end of cell life at the right time. So imagine that there's an abnormal pathway that's like a neighborhood street, and there's lots of options and turns that one could make to get from point A to point B. And imagine that getting to point B is what is kind of the final step towards this uncontrolled cell growth or some block of cell death. So in theory, we have all these potential targets for targeted therapy, so to speak, across this whole pathway. And the problem is that with one targeted therapy, you may have a resistance that'll emerge with an alternate path that can arise. And so if you block this first conceptual node in this pathway, this signaling pathway, in theory, the system might at random sort of develop or amplify this other path that will become prominent and then therefore make the targeted therapy fail after some period of time. And you could block a sequence of pathways, of course, and that might be, again, new kinds of molecules that get invented to do these types of things. And then, of course, you can have a resistance pathway yet again emerge because there's a redundancy of paths that can do that. Thus, effective targeted therapy to treat cancer may require a sequence of targeted therapies that drives a cancer cell's signaling system to a sensitive point where you can kind of zap it at the end after you've given it no other choice. So that's what I'm going to try to represent in these next few slides. You could have treatment that might begin with two targeted therapies, which limit the options of the path that may still lead to uncontrolled cell growth. And when you block those two paths, you'll see that a third path could emerge in the matrix of cellular signaling systems that are again existent in the cell. And then you could add two more targeted therapies, like represented by the boxes 3 and 4, that further drives the cancer-causing pathway to some into some pattern that you could treat, and then you could see that would still result in the pathway moving down the only choice that's available, which is where the pathway will go in theory. And then you'll add more targeted therapies, 5, 6, and 7, and in theory that drives the path down to really only one choice to get to that uncontrolled cell growth. And at that point, you have number 8, the targeted therapy number 8, and in this conceptualization, that would block and leave that whole signaling pathology ineffective. Now, if you just started with targeted therapy number 8, that might work for a little while. We have numbers of targeted therapies that work for a little while and have resistance, and we envision that resistance is kind of the process of again, through randomness and emergence and other kinds of microenvironment influences, drive cellular signaling systems to essentially, quote unquote, find alternative pathways to where uncontrolled cell growth happens. So the future of targeted therapeutics is probably associated with the need to block not only nodes in the pathway involved in tumor development or progression, but the development of targeted therapy needs to also be addressing the surrounding pathway nodes that likely account for resistance. Now, we do want to highlight there are people working on combinatorial approaches for different types of cancer, and many of these approaches use targeted therapies, and others use non-targeted, more generic chemotherapeutic therapies combined with targeted therapy. This is a second example, and I'll follow with the third example. That middle one was melanoma, and this is metastatic renal cell carcinoma. And again, you've seen the word sequential or combination in these, and again, we've had combination therapy for decades, which would mean combination chemotherapy, but we're talking about combination targeted therapy and sequenced targeted therapy, which is relatively new on the horizon. So as we've said, studies in sequential and combinatorial cancer therapies are not new, but studies in sequential and combinatorial targeted cancer therapies are relatively new, and these studies require a deep understanding of cellular signaling. And accelerating the effectiveness of cancer research at this time requires acceleration of the understanding of the cellular signaling systems, and it's likely that computational modeling, as you've heard me say already, is the fastest approach to accelerating our understanding of cellular signaling. So conclusions. First of all, thank you for listening. Knowing about genes and mutations of the cancer cell doesn't necessarily tell you everything you need to know about cancer's diagnosis, treatment, or prognosis. That's what we started with. We know that the existence of epigenetics or non-mutational factors that change gene expression mean that gene sequencing does not tell the whole story in cell biology and is an imperfect way to fully understand a cancer cell. We know that cellular signaling systems likely have an inherent complexity that exhibits emergent behavior and is amplified by interactions with local microenvironment of the tumors. Some of the inherent complexity of cellular signaling systems may be due to the emergence that is related to simple dynamics, as we saw with the two rules and the algorithm on the computer display. Some of the inherent complexity may be due to overlapping components among different pathways, isolating reactions, and redundant pathways and feedback loops. In complex cancers, understanding diagnosis, treatment, resistance to treatment, and prognosis may require deep understanding of cellular signaling. Effective treatment of complex cancers likely requires combinations of targeted therapies used in a defined sequence. And finally, that accelerating progress in this arena likely requires significant resources directed at computational modeling of cellular signaling systems. Thank you very much for listening to these comments, and look forward to your thoughts on these comments.